Session 2 is all about the future of the brain and the future of the mind. If you think the world has changed dramatically in the last 5 years, you ain't seen nothing yet. Things are moving. In this session, you'll hear many challenges of accepted ideas, such as the Red Queen in Alice in Wonderland, who only remembers the future instead of the past. And she challenges our linear conception of time. In session 2, we'll also see some new innovations, such as brainwave control of machines that will allow paralyzed people to walk, memory-boosting brain implants, and machines that teach themselves. So, if Joseph would quiet down. If rats can be trained to detect landmines in Mozambique, then what are the untold possibilities for the human brain? We can look forward to hearing many more of these possibilities in session 2. We're going to start with Baroness Susan Greenfield, and here's Joseph, the troublemaker, to introduce her.
That is a title I'm very happy to assume. Troublemaking, as Bo Lotto said earlier, probably has a lot to do with uncertainty and creativity. And Susan, it was one of the first neuroscientists that I met when I ran the Mind Science Foundation in San Antonio, Texas. We funded consciousness, and she's one of the leading researchers in the field of consciousness. And she stirs things up and is a little bit of a troublemaker herself, being very creative and sort of giving the old boy network in England a little bit of a poke in the ribs. In the meantime, she is a member of the House of Lords of Parliament. She has accumulated, deservedly, 32 honorary degrees, including the Légion d'honneur, that's Texas French, founder and CEO of NeuroBio, and a member of the Royal College of Physicians. Please welcome Baroness Susan Greenfield, my good friend.
Oh, good afternoon, everyone. It's a huge pleasure to be here. And Joseph and Glenda, thank you so much for inviting me over. Because time is so short, I'd really like to say what a delight it is to be here and what fun I'm having, but I ought to cut to the chase. And what I want to do is cram into this very limited time, if you like, a potpourri of a neuroscientist's view of the various issues relating to the mind in the 21st century. So what we're going to look at, albeit at lightning speed, and I'm going to do this in parallel, is how we can blow the mind. And one day I'm going to show this picture, and the guy will be sitting on the front row. I'm sure he's a college professor somewhere now. We're going to look at expanding the mind. This is George Bernard Shaw, and clearly he's obviously very thoughtful and conscious. And then finally, we're going to talk about losing the mind, something that's dear to all our hearts, and we've heard about from very eloquent presentations this morning, which is Alzheimer's disease. So that gives me about 7 minutes on each of these topics. So here we go. This reminds me of when I first dissected a human brain. I'm sure many of the scientists here did that. I remember thinking, if I didn't get a bit of tissue, if I wasn't wearing gloves and I got a bit of tissue under my fingernails, would that be the bit that somebody loved with? Would it be a memory? Yeah. Can you have an emotion under your fingernails? Can you have a habit like biting your fingernails under your fingernails? Sorry to do this to you after lunch. There we are. Yeah, so that reminds me. Now, what we're going to do then is, first of all, approach the issue of what a mind might be, not some airy-fairy philosophical alternative to the squalor of the physical brain. I hope I'm not offending any philosophers here. But rather, I want to persuade you that we scientists, we bovine scientists, we too can have a view on what the mind might be. So the brilliant thing about being born a human being, unlike a goldfish, say, which frankly doesn't have great personality, does it, is that you are destined to have a great personality, and you're going to be unique. And for the 100,000 years we've stalked this planet, no one is going to have a brain, or rather a mind like yours. Now, why is this? It's because this is how you started life. There we are. in the first 2 years of life, you can see that the human brain postnatally is characterized by astonishing increase in brain cell connections. Although you're born with a fair complement of brain cells, it's the connections which characterize your brain and distinguish you even from a clone, that's to say your identical twin. Why is this so special? Well, it means you are able now to do what a goldfish can't do, and that's adapt to your environment. And that's why we occupy more ecological niches than any other species on the planet. We don't run fast, we don't see well, we're not strong, but gosh, what we do, although others do it to greater or lesser extents, we do it superlatively, we adapt to the environment. And this I'm assuming Dr. Deutsch will talk about later, which is so-called plasticity of the brain. It doesn't mean, of course, that the brain is made of plastic. It means from the Greek plastikos, to be molded. Now, what do connections do for you as they are formed? Well, I'd like to suggest that they give you a meaning over time. A baby confronted with this might think it's a gold shiny thing, might want to put it in its mouth, it won't mean anything. But as the child interacts and has experiences that are then reflected in the laying down of the connections, it will remember and learn that something goes on fingers. And a gold shiny thing only goes on a certain finger. And it only goes on a certain finger under certain circumstances. Then they might acquire one of their own, and that particularly otherwise generic-looking object will become perhaps the most special thing to them. Then they may go from the honeymoon to the divorce. It might become the most reviled thing, triggering the most amazing sensation. Why? It's just something a kid would want to put in its mouth. And yet, what's changed? Not the thing. No, it's the connections that have changed. Similarly, if I'd come on today dressed like this, this is, by the way, an English ghost, in case you haven't seen one recently. I doubt if anyone here would be very frightened, but someone with dementia or a small child might well be frightened. Why? Because they take it at face value. They haven't got a framework of previous experiences to understand, to go beyond the sensory press, that this is someone simply dressed up. And because it's novel, it's potentially life-threatening, and therefore you're frightened. So, here you are living your life. This is downtown Oxford. And everyone, this is how we still are, and everyone is just like everyone in this room. You have a unique trajectory in space and time. If you like, it's a life story. It has a beginning, a middle, and hopefully in the dim future, it will have an end eventually. But it's that linearity, that sequence of beginning, middle, and end that is your life story. It's your identity. And which is why I think stories generally have a resonance, and we can think about that a little bit more. It also echoes the thought process itself, which is, after all, a sequence, is it not? That's what distinguishes it from a feeling. Anyway, so here you are living your life, and as you live your life, you're evaluating everything that happens to you in terms of what's happened before, and in turn, that ongoing experience will upgrade and change your ongoing connections. So you're in a beautiful dialogue with the outside world, as shown by this rather clunky transition here. So I'd like to suggest to you that the mind is not an alternative, some airy-fairy philosophical alternative, as used to be thought of mind-brain dichotomy. No, the biological basis of the mind, I'm suggesting, is the personalization of your brain through unique dynamic configurations of your neuronal connections, driven in turn by your unique experiences. Now, if you buy into that, then, let's go on to the three things. How do we blow the mind? What do we mean by that in neuroscience terms? Well, let me introduce you to an important area of the brain called the prefrontal cortex, and this actually came up over lunch. It's shown here in turquoise. It's very dominant in the human brains and becomes decreasingly less important as we descend the evolutionary scale. Suffice it to say, it's not the center for this or that, but it does connect up with many, many brain centers. And as has been shown in biology quite a lot, where individual development reflects evolution, the posh way of saying that is ontogeny reflects phylogeny, so we find that this area of the brain is underdeveloped up until late teenage years, early 20s. So those of you who have teenage kids might start to recognize some of the traits that I'm about to explore. Interestingly enough, schizophrenics also have a less than functional prefrontal cortex, and we see various similarities. Both children and schizophrenics are easily distracted, both have shorter attention spans than adults, neither can interpret proverbs. If you say to a schizophrenic, what does it mean people who live in glass houses mustn't throw stones? Oh, sorry, I've forgotten to show you the thing here. That's the prefrontal cortex, and here we are, we're on to this one now. They can't interpret proverbs, and both have an underfunctioning prefrontal cortex, so they're sort of living in the moment. And here you are, that's a schizophrenic brain scan showing the underfunctioning prefrontal areas. Other people, interestingly enough, with underfunctioning prefrontal cortices are less obvious. People with a high body mass index, that's to say people who are heavy relative to their height. And also, and these are true research papers, you can see them quoted here, such people also are more reckless in gambling tasks. Yeah, now just let's think that through, what that might mean if we're thinking about the prefrontal cortex. Okay, so anyone who eats knows the consequence of eating, but the thrill of the food, for some more than others, trumps the consequences that you're going to put on the calorie, put on the inches. Anyone who gambles knows the consequence of gambling, but the thrill of the roulette wheel or the roll of the dice trumps the consequences. You're probably going to lose your money. How does that relate to schizophrenia? Well, this is a series of paintings, very famous by schizophrenia, that gives us a clue. Everyone would recognize that as a cat, but by the time we get to bottom right, no one would recognize that as a cat. So what do these things have in common? I'd like to suggest to you that in all cases, the thrill of the moment is trumping the consequences. Okay? So the senses, therefore, are dominant. Given that, we could attempt, therefore, a rather interesting division in how the brain works or the mind works. We can call it the meaningless mode, where the prefrontal cortex is underfunctioning, as a contrast to the meaningful mode, where the prefrontal cortex is fully active. So this would be characterized by strong feelings as opposed to thought, by sensory simulation, like eating and so on, as opposed to cognitive, assigning meaning to something. Living in the here and now, think of the gambling and the eating. Living with the past, present, and future, think of your life story. External input will be dominant. Here, internal perceptions will dominate. This has little meaning, think of the wedding ring. Here, personalized meaning, think of the wedding ring. Here, a reduced sense of self. You're not self-conscious necessarily. Here, a strong sense of self. Here, no time or space. We know that when the area is damaged, you can get something called source amnesia, where you don't remember episodes, you just have a kind of generic memories. Here, a clear space-time reference. This is mainly infants and children, which is why those of you who are parents to teenagers might recognize things on the left, as opposed to older children and adults. And interestingly enough, that much overworked chemical, I'm afraid I'm going to have to bring it into play too. In teenagers, you have the biggest surge of dopamine you'll ever have, as opposed to elsewhere. So this is how it's always been. And I think that what's very interesting is that although this is the characteristic of the younger brain, nonetheless, we can look and see how it might operate under other circumstances. For example, we know that dopamine will inhibit the prefrontal cortex that we've just looked at. So if you do anything that is stimulating dopamine, then you're going to swing over to the left-hand side, possibly. So that will mean, if you're very excited, all drugs of addiction will release the drug dopamine. And in case you haven't had a reward recently, this is what they look like. Rewards are related to the release of dopamine. Okay, so moving on very quickly, it's ever been thus. This is from a vase showing the Bacchae by Euripides, a famous ancient Greek playwright, where this poor man in the middle, he's called King Pentheus, is about to be torn apart by these wine-crazed women worshipping the god Dionysus. So it's always been the case that we've done wine, women, and song, drugs and sex and rock and roll, and they all have something in common. I would suggest an abnegation of the sense of self, a swing to the sensory, at the expense of the cognitive. We talk about having a sensational time. Who's going to come out now and have a cognitive time? No one. No one's going to do it. No one. We talk about letting yourself go, don't we? And here we are, you're letting themselves go. Here, you know, the white water isn't standing for anything. It doesn't mean anything. It's just there. Or you can have, this is from Australia, where the selling point was to be totally out of control, notice. Yeah. So we seek out and always have times in our lives when we perhaps reduce the activity of the prefrontal cortex in favor of living for the moment and sensation. And my concern is that perhaps in this video game world, what we're going to be doing is tilting the balance too much. Right, moving on to the next thing. We're now going to, having solved that, do consciousness and expanding. And this is from our own work. What I find very interesting about consciousness, people talk about it being all or none, but actually if you look at the two most obvious states of unconsciousness, namely sleep and then also anesthesia, in both cases they are graded. And it struck me that if unconsciousness can be continuously variable, as both these very familiar conditions are, what if consciousness was variable? You could have more of it or less of it from time to time. And if you think about it, you're talking about raising our consciousness or deepening our consciousness. It doesn't matter which way you go, but you know, we're having more or less. And indeed, we can think of this kind of scenario where, therefore, we should be in search of a process where consciousness is continuously variable. And in the great manhunt that's on for looking at what's called a neurocorrelative consciousness in the brain, what I'd like to suggest is on the shopping list should be a process that varies in degree from moment to moment. So that means we rule out static brain regions and activity in brain regions. We will we rule out single cell, generating their little electrical blips that are all or none, and we've looked for something in between. And what we have worked on in my lab, and what we have developed using very special dyes that give an instant readout in real time of what the brain is doing, are these so-called neuronal assemblies, which are large-scale groups of brain cells that occur within less than a second and involve greater than 10 million cells. So it's a bit like a stone in a puddle, if you like. If you think that we're looking in the brain for something, a trigger, the stone, that when it's activated, thrown, will cause a highly evanescent but nonetheless very excursive degree of ripples that are highly transient. So what we're going to do is use this metaphor of the stone in the puddle to try and explore whether these neuronal coalitions, these assemblies, could fit the bill. This is what they look like. This is from my lab, and as you can see, these are 1 millisecond frames showing that we are on a timescale commensurate with neuronal activity. Okay, so if we study this in relation to consciousness, then it can answer various questions. For example, why does an alarm clock wake you up? Well, that's like a very forceful stone being thrown. And so we can talk about the force of the stone. Next, how can personal experience affect your consciousness? Well, this is me a few years ago with some colleagues, and I'm deliberately showing it because it won't mean anything to you. It means a lot to me. And I'm sure you could have similar photos. And this is due to the size of the stone. That's to say the neuronal connectivity that has been forged by those experience. Third, what happens when you're dreaming? Weird type of consciousness. Well, that is where the stone isn't very forcefully thrown because the input is not coming from the outside senses. Fourth, what about arousal, making you excited? Well, these rather technical pictures on the left are chemical fountains, of which dopamine is one, that will modulate or make it easier or harder for the ripples to spread. We could call that the viscosity of the fluid. Next is how do anesthetics work? Well, again, this is from our own lab, showing that assembly is sensitive as you are reducing the possibility of consciousness, so you see we're reducing the assembly size. So that's acting on a holistic assembly. Next, what is animal consciousness? I love this picture. Normally that should bite the lady or run away. And we like this because I'm sure many of you have cats or dogs, and yet I'm sure you'll say they're conscious, but they're not quite like we are. Now you can get around that by saying, yes, an animal is conscious, but not as conscious as we are. So a dog is conscious, but not as conscious as a human. And a rat is conscious, but not as conscious as a dog. So it's very useful to think of assemblies like that. Next, how can we differentiate hearing and vision? Because by the time they've been turned into volleys of action potentials by the cochlea or the retina, they're treated the same by the brain. So why do we have the subjective difference that is so distinct with hearing and vision? Well, these are assemblies. You can see here the patterns showing the red blobs in 11 different experiments. All you have to see is the red blob is to the left, and with the auditory system, the red blob is to the right. So we can, using assemblies, differentiate these two sensory modalities, but we can only do so at 300 milliseconds, as shown here, showing that that's usually the epoch for a window of consciousness, showing that that might be related to it. Finally, why is pain perception so variable? Well, I'd like to suggest to you that the size of these assemblies will determine how much pain you feel. Very briefly, pain is expressed in associations, pricking, stabbing, burning. There's a diurnal threshold for pain. If you want to go to the dentist, go at lunchtime. It's when your pain is less, and that's probably due to those modulators. If you anticipate pain, it's greater because you're building up all those connections. You're probably familiar with people who have had amputations who will still feel pain in the severed limb because of the feedback that's not there triggering the assembly. Anesthetic action can now be explicable because it's reducing the size of the assemblies, and otherwise people haven't been able to really understand how anesthetics take away consciousness. Morphine, obviously a well-known painkiller, the person says they feel the pain, but it no longer matters. And we know that the morphine works on naturally occurring targets in the brain that will reduce the size of the assembly. When people have morphine, they say they have a dreamlike euphoria, which I've said is like a small assembly. So that's really a kind of gallop through. What I haven't been able to do, I love this picture, look how people see male scientists, by the way. So on the left, you've got for on the right, and then in the middle, a miracle occurs, and the guy says, I think you should be more explicit here in step 2. I often show this when I talk about women in science to show people's perception of scientists, by the way. It's quite interesting. But the whole point is that what I've shown you is a correlation, not a causality. I can't say how the water of the clung combined of neurons is converted into subjective experience. But we are starting now using this approach to see how closer and closer they can match up. So a degree of neural activity is intensity of the senses, extent of pre-existing associations we've looked at, we could call significance, availability of modulators, arousal, and indeed hormones could also do that. Formation of competing assemblies, like in fast-paced sports, would be a distraction. Input from the prefrontal cortex, which we've looked at, would give you the narrative. And therefore, we can start to build up the kinds of ways we might interpret an assembly. This is a very busy slide, which I'm not going to go into details, showing how mind, brain, and consciousness can all connect up. Very briefly, we have a personalized system that we saw was the mind, and that will act as the stone in the puddle for triggering within the brain these assemblies, which in turn on the right will correlate with degrees of consciousness, all of which very conveniently aligns with the different disciplines that we associate traditionally with those things. Okay, so very briefly then, on to Alzheimer's disease, losing the mind. And this is work that we're doing in Neurobio, my company, and this is, again, just a taster. I thought today I'll just give you a taster of all the different things neuroscience can do and why it's such fun. Okay, so after the very eloquent description of Alzheimer's we've heard previously, that's marvelous because I now can continue that story. What's very interesting about Alzheimer's that people ignore, they tend to look at amyloid and tangles and this tau because that will be the characteristic of the postmortem brain. But in fact, we know that really Alzheimer's starts 10 to 20 years before the symptoms appear, and only spread to the cortex, the outer layer, in the later stages. So, oh, sorry, I keep missing my buttons here. Okay, so here you see a paper from the Bracks showing the spread of the Alzheimer's. But I'd like to draw your attention more to the top left, where that black triangle is. That's the brainstem, and it's there that we should ask, why is it that cells start to die there? Because Alzheimer's disease, I want to repeat this, is not a generic feature of neurons. Not all neurons will degenerate, so we need to find out what the special feature is, and then we can obviously exploit that and tackle it and intercept it. So let's look at the brainstem then, what's so special about it. Well, this is from a review written a while ago showing most cells in the brain have various properties, but what's very different about the brainstem ones is, you don't have to worry too much about all these technical anatomical labels, is that they come from a different part of the embryo. You can see there, 3 dots down, it says alar plate rather than basal plate. That means at a very early stage, at about 4 weeks gestation, already these cells are showing their differences. And what's very exciting about their differences, when they finally elaborate into the full brain, is they maintain their ability to grow again. And now that surely would be a good thing. Well, the problem is it wouldn't be a good thing because we know that you can inappropriately activate the cells so that the trigger, which is calcium going into the cell eventually, will be toxic in mature cells. So one has a sort of Jekyll and Hyde situation where the same chemical that is fantastic in development, actually in the context of the mature brain, is inappropriate. So here we have in the normal adult, most cells will stabilize, and then if you have damage, you'll have compensation by the extant cells. Whereas if those brainstem cells are damaged, what will happen, because they've retained this embryological feature from their provenance long ago, at 4 weeks in the womb, now what will happen is they'll mobilize a molecule that will signal developmental processes, which actually will be toxic, so that will set up the terrible cycle of cell death that we call neurodegeneration. So what we need to do is intercept that, and to cut a long story short, what we've done is identify the evil chemical, which is a peptide. That's just a fragment of a protein. And here you can see that our theory is correct. In the rat brain, as the rat is getting older, this evil chemical is declining. It's very high in the young rat, but this is mirrored in the post-mortem Alzheimer brain, where in the healthy adult, well, they're not healthy anymore, they're dead, obviously, but compared to the Alzheimer's, you can see it's almost doubled in amount. We can also show that it's a potential biomarker. Here you can see in the cerebral spinal fluid, that's the fluid that breaks the brain and spinal cord, rather disparagingly referred to as the urine of the brain. You can nonetheless see that the levels are much higher of our evil chemical, both shown there on one gel and also as a histogram. We know that this fits into the picture of amyloid and tau because the peptide will actually drive the production of amyloid and tau. So I don't want to sound too dismissive of amyloid and tau, it's just I don't feel it's the primary target, which is why no drug targeting it has actually succeeded since 2002, because although it's part of the picture, it's not the primary driver. Moreover, this peptide derives its own target. You can see here the toxic peptide has got a greater green ring around it. So it not only is doing all this, but it's creating its own target, so-called receptor. So there's a feed-forward mechanism going on. So it creates its own target. So clearly, just to recap, we think that neurodegeneration is an aberrant form of development. We've shown it's increased in the Alzheimer brain, it's increased in cerebral spinal fluid, it drives production of amyloid and tau, and to make matters worse, it creates its own target so it can do even more damage. Obviously, we want to intercept it. Now, the problem with all drugs, in my own view, and why things perform well in mice running mazes, but not in the clinic, is because the big difference is in the clinical situation, the brain is already flooded with our evil chemical. That will be occupying the relevant receptor sites, the target sites, so that poor little man-made or woman-made chemical doesn't stand a chance against the naturally occurring agent, which is already there. So what you need to do is develop something where you fight like with like. And what we've done, and forgive me for this technical picture, is take the evil chemical and bend it into a circle, so it's called cyclization, so that it can no longer be active, but it will still compete with the naturally occurring chemical. It's called NBP14, neurobiopeptide 14, because it has 14 building blocks or amino acids. Okay, so using that in 3 different tests, you can see very quickly, because Joseph's hovering, you can see the 3 different tests we've used of calcium going into the cells dying, and compensatory release of the parent molecule. I hope you can see on the bottom, the evil chemical has caused calcium to go in, it's compromised cell viability, it's triggered a compensatory release. In all cases, that's blocked with the drug. Amyloid comes along behind, it's also had similar effects, and the amyloid too is blocked by our drug. So, where does that leave us in the long term? Well, the long-term goal is twofold, really. One is to develop a biomarker, because we know that we have this 10 to 20 year window, which if we had a direct readout, not of the probability of cells dying, but the fact cells are actually dying, if you could see that, then you could intercept it early, and you could even start people on existing drugs early. So that in itself would be wonderful if you could have a biomarker, a blood test that all of us over a certain age went and got, rather like you might have a cholesterol test. The next stage is also to have a treatment that stops any more cells dying, not that reduces. All the drugs do at the moment, like galantamine and Aricept, they slow down the process. The current heralded drug by Lilly that's just coming out apparently just slows things down. That is not good enough. We need to stop the cells dying. And if this is a candidate, then that would be great, but we're trying to get a smaller molecule and doing medicinal chemistry. And so even if you had the symptoms, if you knew they weren't going to get any worse, that would be a great advance. Now put those 2 things together. If you said to someone, well, I'm afraid there's good news and bad news. The bad news is you've got an elevated marker in the blood. You haven't got symptoms now, but I'm going to this chart, because we can read it off. In 5 years' time, I'm afraid you're going to start feeling a bit confused and have memory loss and so on. But the good news is we now have a medication that you can take that stops any more cells dying. So take it now. Take the medication now, even before the symptoms come on, and then the symptoms will never come on. So it would be an oral medication and accompanied by a routine blood test. And so that's what we're trying to do and to develop. It's a long way off, but you can see, I hope, that the goal is very clear, and I hope it's a realistic one. So to summarize then, the mind means many things to many people. We can think about the impact on younger people and video games. We can think about the neuronal basis of consciousness and how we can put ourselves in environments to expand our consciousness and what it might mean. And it touches on the real frontiers of philosophy. And perhaps on a more practical level, so that we can enjoy doing all those things, it would be nice if we had an effective treatment for preserving not just long age, but a healthy mind in it. Thank you very much. Okay.